How do dormant lung cancer cells reactivate? Imagine a tiny seed lying dormant in the soil, waiting for the right moment to sprout. That's similar to how some lung cancer cells behave after initial treatment or during metastasis. These cells can enter a state of dormancy, where they stop dividing and seem to disappear. But they are not gone forever. Over time, certain changes in their environment can wake them up, leading to cancer recurrence. One major factor is the microenvironment around these cells. The extracellular matrix, which is like a scaffold of proteins and fibers surrounding the cells, can change over time. When the matrix remodels, it can send signals to dormant cells, telling them it's safe to grow again. For example, neutrophils, a type of immune cell, releases DNA and enzymes that can alter the matrix. This process activates signals in dormant cells, prompting them to re-enter the cell cycle and start dividing again. The immune system also plays a complex role. Initially, immune cells help keep cancer cells in check, but chronic inflammation or stress can shift this balance. When immune cells like neutrophils are activated by stress hormones or inflammation, they release proteins that can stimulate dormant lung cancer cells. These proteins can cause oxidative stress and activate specific pathways that encourage the cells to wake up and grow. Stress hormones such as adrenaline and cortisol are also involved. When a person experiences stress, these hormones can activate immune cells, which then releases factors that promote cancer cell reactivation. This creates a microenvironment that favors tumor growth, even long after the initial cancer treatment. Inside the dormant cells, metabolic changes are key. These cells adapt their energy use, increasing fatty acid oxidation and producing reactive oxygen species. These shifts activate certain transcription factors that help the cells restore balance and prepare to divide again. Pathways like Notch4, Hedgehog, and AF6-SEGF are also involved in signaling the cells to escape dormancy. Epigenetic reprogramming is another important piece. These are reversible changes in gene expression that don't alter the DNA sequence but can turn genes on or off. When environmental cues change, dormant cells can switch back to a proliferative state by modifying their epigenetic landscape. Interactions with other cells in the tumor's neighborhood also matter. Cells like macrophages, fibroblasts, and endothelial cells secrete growth factors and cytokines that can encourage dormant cells to reactivate. These signals help create a niche that supports tumor growth and spread. Understanding these mechanisms is vital because dormant cancer cells can survive initial treatments and later cause a relapse. Recognizing how microenvironmental changes, immune influences, metabolic shifts, and epigenetic reprogramming contribute to reactivation helps researchers develop better strategies to prevent recurrence. It's important to remember that each person's situation is unique, and ongoing research continues to explore ways to target these pathways. If you or someone you know is dealing with lung cancer, always consult healthcare professionals for personalized advice and treatment options.